Great. Thank you very much for having me. And what I'd like to do this morning is talk a bit about the fluorescent proteins and some of the new variants that are available for the work that we are doing, and then show you an example of how we've used FRET-FLIM to analyze the interactions of proteins inside the living cell nucleus. In the images I'm showing on this first slide, this is the nucleus of a cell, and these are regions of heterochromatin within that cell. And I'm going to talk to you about how we've used FRET-FLIM to look at the interactions of proteins within these regions of heterochromatin. So I'll start out by talking about the general characteristics of the fluorescent proteins. I'll tell you a bit about some of the Aequorea Victoria color variants that came from the original GFP, and then talk to you very briefly about some of the new fluorescent proteins that have come out of corals and fish. And then we'll talk about how we use these fluorescent proteins in FRET-FLIM to actually monitor the interactions between nuclear proteins and living cells. And I'm going to focus in on a transcription factor called EBP alpha and a heterochromatin remodeling protein called HP1 alpha. So let me start out by talking to you a bit about the general characteristics of the fluorescent proteins. And as I'm sure you all know, the jellyfish Aequorea Victoria is where GFP came from. And this jellyfish makes a chemiluminescent protein called Aequorin, which is a calcium binding protein that uses cilantrazine as a substrate to generate light when calcium binds, and it emits blue light. So the jellyfish has also evolved a second protein, GFP, the green fluorescent protein, which is an autofluorescent protein that absorbs that blue light energy and then shifts that emission to green light. And this is actually a FRET process that takes place inside the jellyfish. And of course, the cloning of GFP caused a revolution in cell biology because for the first time, it enabled us to genetically encode fluorescent labeling. So again, as I'm sure you know, in 1996, the crystal structure of the jellyfish green fluorescent protein was solved, and it showed that this GFP adopts this absolutely wonderful 11-stranded beta barrel structure. And in the center of that beta barrel is a cyclic tripeptide that is the chromophore for GFP. And this beta barrel has to form in order to position this chromophore within the center of the barrel. And so that explains why you need the entire protein in order to be fluorescent. So very early on, investigators realized that this protein that's adapted for the cold waters off the northwest coast of North America behaved very poorly inside living cells. It doesn't like to be at physiological temperatures, so improvements were needed. So among the first things that were done by investigators were to humanize the codon usage within the jellyfish green fluorescent protein at a optimal initiation site, and then begin to play with the amino acids that are in and around the chromophore and find out which ones improved its maturation at 37 degrees. And so as a result, a number of mutations were introduced into the beta barrel and the chromophore itself, resulting in what we refer to now as the enhanced FPs. So when you hear EGFP or ECFP, we're talking about proteins that have been optimized for expression in mammalian systems. Now, most of the fluorescent proteins that are found in nature are either obligate dimers, tetramers, or higher order complexes. This is simply the way that these fluorescent proteins form in nature. The Aequorea GFP was unusual in that it actually could be crystallized as a monomer. And this is a good trait. So in living cells, typically we don't see dimerization of the enhanced FPs, provided they're free to diffuse. Now this is an important point because if you express the fluorescent proteins in a diffusion limited volume, such as membranes or inside organelles, then dimerization can and often will result. So Roger Chen's group recognized this problem and again did mutagenesis and determined that a simple substitution of an alanine with a lysine at position 206 generated what we refer to as the monomeric versions of these fluorescent proteins. And so if you're interested in doing any FRET-based measurements, regardless of whether you're doing it in membranes or where they can diffuse, it's an important point to be using these monomeric versions of the FPs. So now I'd like to just briefly talk about the Aequorea color variants. So this is the spectra for the wild type GFP from the jellyfish, showing that tricyclic chromophore that forms in the middle of the beta barrel. And it had a fairly complex excitation spectra with a peak around 400 and a second peak around 480, and it emitted around 513. And so Roger Chen's group figured out that if they substitute this serine position of the chromophore with a threonine, they stabilized the chromophore, and that yielded a single excitation peak at 488, with the emission still being around 513. And this was actually a brighter variant. And so this was the first evidence that by changing the sequence around the chromophore, you could dramatically change the spectral characteristics of the FP. So the S65T was the first of the optimized FPs. And by doing other substitutions in and around the chromophore, they were able to produce blue, cyan, and yellow color variants of the Aequorea GFP. So now what I'd like to do is I'm just going to briefly mention some of the more optimized versions of these FPs. So one of the best of the eGFP variants currently available is mClover3. It was taking eGFP, doing mutagenesis to generate clover, improving it, and improving it again. And what you end up with is a very bright green fluorescent protein that has very good maturation characteristics. It's very photostable, and it's very useful as a FRET donor for the RFPs. I'm going to refer to intrinsic brightness as a way of comparing the FPs. Intrinsic brightness is simply the quantum yield times the extinction coefficient. And so original eGFP has an intrinsic brightness of 35. Clover3 has an intrinsic brightness of 85. The cyan fluorescent proteins that came out of the early mutagenesis screens have been extremely useful for FRET. And so the original eCFP had a number of problems associated with it. So Dave Piston's group did mutagenesis to generate cerulean, and then a number of other groups have taken cerulean and gone on to do mutagenesis screens to generate turquoise, turquoise-2, and cerulean-3. Each of these is a very bright cyan fluorescent protein, so intrinsic brightness in the blue range of 28 is a bright blue fluorescent protein. The mutagenesis improved the photostability. Importantly, for what we're going to talk about later, the cyan fluorescent proteins have a single component lifetime, which is why we prefer to use these variants in lifetime measurements. Also, yellow fluorescent proteins came out of the early mutagenesis screens, of which M. venus and M. citrine are among the best. They're still very useful, even though they were generated nearly 20 years ago. A number of mutations were introduced, but the key mutation they found was a mutation in the beta barrel that actually contacts the chromophore. This yielded a very bright yellowish green fluorescent protein with an intrinsic brightness of 54. It matures rapidly. Importantly, Venus is not terribly photostable, but this is an important characteristic. You can use it for things like photobleaching or acceptor photobleaching FRET. So if you need to use a protein that you're going to bleach, this is a good one to use, and they're excellent FRET acceptors. So the Aequorea mutations generated, as I mentioned, blue, cyan, green, and yellow fluorescent proteins, but this was as far as investigators were able to get it towards the red spectrum. So there was still a need for improved color variants. And so a number of investigators have been screening corals and fish and other marine organisms looking for novel fluorescent proteins. One of the first ones that came out of these types of screens were the red fluorescent proteins. This was work done by Motz in Moscow, and they used reef corals and began to look for novel fluorescent proteins. And this is Discosoma striata. It's the mushroom anemone. And in Discosoma, they were able to identify 4 distinct fluorescent proteins, of which the one on the bottom here is the one that has been most universally used. That is DsRed. And so DsRed has a distinct fluorescent spectra from the Aequorea FPs. It's easily detected with standard optical filters. And because there's much reduced cellular autofluorescence at these longer wavelengths, it was really a useful tool for cellular imaging. However, there were some distinct problems with DsRed. As I mentioned, these FPs like to form higher order structures. And DsRed is an obligate tetramer that takes nearly 20 hours for it to fully mature in mammalian cells. And it also goes through a green intermediate as it's maturing. So this is a real problem if you want to use it in multispectral imaging. The other problem was it forms oligomers, so it tended to misdirect any fusion protein you made to it. So to overcome this, Roger Chen's group went in and used directed mutagenesis to break the tetramer, but they ended up with a non-fluorescent protein. And so they then did random mutagenesis of that non-fluorescent protein, bring back the red fluorescence. And this is where MRFP1 came from. The advantages of MRFP was, of course, it's a monomer. So this overcame the issue of tetramer oligomerization. It got around the problem of slow maturation. It matured very quickly, and its emission spectrum was actually shifted deeper into the red, which was another nice characteristic. However, it's a monomer, so it had decreased quantum yield compared to DsRed, and it had decreased photostability. The reason they like to form tetramers is for photostability. And the big problem with MRFP was this issue right here, that as much as 60% of the protein formed a non-fluorescent form that absorbed but did not emit. So this was a real problem for MRFP. So we still needed improved FPs. And so again, a number of groups, including the Chen group, took MRFP, and then they did multiple rounds of mutagenesis on it to try to find additional novel fluorescent proteins. And this is a method called directed evolution. So they simply did random mutagenesis and used a FACS sorting to identify new fluorescent proteins. And from here, we got the fruit series of fluorescent proteins. Very quickly, we had a whole set of new fluorescent proteins. And so this is honeydew, this is banana, orange, tomato, this is dimeric tomato, tangerine, strawberry, and cherry, and these are the emission spectra for all of them. So in a very short period of time, we had a whole set of new fluorescent proteins. Of the fruit series, really only M cherry and tandem dimer tomato are broadly used. The other ones had a number of problems associated with them and not used frequently. For example, M orange was shown to photoconvert to a red form by simply observing it. So this is some of the limitations of these novel FPs. But importantly, what came out of this was structural information about how to make different color fluorescent proteins and the photophysical properties of those fluorescent proteins. And so still, as I mentioned, some of these were not terribly useful. We still needed additional FPs. And so a number of groups have been looking at other corals and other marine organisms. And one of the more useful ones to come out of these screens was the teal fluorescent protein, MTFP. This came out of this Clavillaria coral, and it's a bright blue-green, teal-colored FP. It has the structure of a cyan fluorescent protein, but it has the chromophore of a GFP. And these are the key mutations that generated the teal fluorescent protein. It has an intrinsic brightness of 54, so it's very bright. It's very photostable. But one problem with teal fluorescent protein is also very acid stable, which means it takes the cell a long time to break it down. So it actually accumulates in lysosomes. So that could be a real problem for background if you're using it. However, if you're interested in studying lysosomes, it's an ideal protein for that. Another one that came out of screen in a lancelet fish was a protein called DLAN YFP, a very bright YFP. It was a dimeric form, so additional mutagenesis was used to generate a bright monomeric form of this protein that's called neon green. And neon green is exceptional because of its intrinsic brightness, an intrinsic brightness of 92. It is the brightest of the monomeric fluorescent proteins. It forms very rapidly, it's very photostable, and it has been used as both a FRET donor and a FRET acceptor. And finally, I just wanted to mention mScarlet. mScarlet was taken a directed evolution approach based on mCherry to generate a very bright monomeric red fluorescent protein. And there are a number of mutations that were introduced into cherry to generate this. I won't go through them all, but it has an intrinsic brightness of 70. Remember, the autofluorescence is extremely low at these wavelengths, so it's very bright. It forms rapidly, it's very photostable, and it's been used as an acceptor for both neon green and clover, among others. So I wanted to finish this section simply by hitting on a couple of key points. There's a reason why investigators have spent a lot of time optimizing these FPs. And so they're optimized for function as protein tags. So if you can at all use the most recent of the FPs, it's wise to do so. They've been engineered to be monomeric. They are rapid and efficient maturation. However, if you're going to use these in the types of studies that I'll describe to you in a moment, you have to ask these critical questions. You have to ask, does the fusion protein to these different FPs behave just like the endogenous protein? And these can be very difficult experiments to do. And the other key question is, does it have all the functions that that endogenous protein has? And again, we can show, for example, this is a histone protein tagged with EGFP and showing a cell going through a complete cell division, showing that these histones tagged with GFP have the normal function. So these are very important things you should be considering if you're going to do these types of experiments. So now I'd like to move on and show you how we've been using some of these FPs in our studies of FRET-FLIM to look at nuclear protein interactions inside the living cell nucleus. I'll briefly talk a bit about FRET and FLIM. You've already heard everything from Dave and Enrico, but I just want to touch again on the use of FLIM to measure FRET. So as you're aware, one of the key issues with FRET studies is spectral bleed through. And so here I'm showing the excitation and emission spectrum for a cyan fluorescent protein, the excitation and emission spectrum for a yellow fluorescent protein to show the spectral overlap between these fluorescent proteins. So spectral overlap is necessary for FRET. The more your donor emission overlaps the acceptor excitation, the more efficient FRET is going to be. And so if you're going to excite your donor and look for FRET in your acceptor channel, there are spectral bleed through components you have to be aware of. For example, no matter where I excite my donor, I'm going to get some direct excitation of the acceptor, that's shown here. And no matter how I set my acceptor channel, there's always going to be donor bleed through into that acceptor channel. And this is the donor bleed through. And these two components have to be accounted for if I want to measure FRET by sensitized emission. However, the beauty of FLIM, of course, is that we don't measure the acceptor. We don't even have to know much about the acceptor at all. We simply measure the effect of FRET on the donor. So our measurement is in the donor channel. And provided I've set that channel properly, there is no spectral bleed through in the donor channel. So fluorescence lifetime measures the effect of FRET on the donor, and there's no spectral bleed through in that channel. Of course, you all know what lifetime is. It's the average time a population of fluorophores spend in the excited state before returning to the ground state. And so the important thing to remember about lifetime is it's an intrinsic property of the fluorophore. And so every fluorescent probe is going to have its own intrinsic lifetime. So for example, fluorescein has a lifetime around 4 nanoseconds. HPTS is a dye we often use as a calibration dye, has a lifetime of 5. Cerulean fluorescent protein actually has a 2-component lifetime of around 3. Coumarin-6 is another dye we use as a standard, has a lifetime of 2.5. Now, if I had a mixture of all 4 of these probes, their spectral overlap would make it impossible for me to know which of the probes was there and how much was there. However, based on their lifetimes and lifetime measurements, it would be possible for me to identify each of these components. Not only that, I could identify how much of each component was present. So I can separate these overlapping spectra by lifetime. And so the beauty of these lifetime time-resolved measurements is that they're unaffected by variations in probe concentration or changes in excitation intensity. So they're ideal measure. It's an ideal way of measuring FRET. And of course, again, as you know, FRET is a quenching event that is a different way for a fluorophore to get down to the ground state without emitting a photon. So this is a probe that's being excited, it goes through internal conversion, relaxes back down to the ground state and emits a photon. That's fluorescence. If there is another probe in its environment that is able to capture some of that excitation energy and relax back down to the ground state, then it's going to quench this fluorescence. So FRET is a quenching event, and that's going to change the fluorescence lifetime because there are now two pathways for that probe to get down to the ground state. So FLIM is a way of precisely measuring that change in lifetime that occurs due to FRET. And as I think you've probably already heard, to measure FRET by FLIM, you really only require two measurements. What you need to know is the lifetime of the donor in the absence of the acceptor. This is the lifetime of the donor. And then you have to measure the lifetime of the donor in the presence of the acceptor. And FRET efficiency is simply the ratio of the lifetime of the donor in the presence of the acceptor divided by the lifetime of the donor. And again, as you all know, there are two different methods of measuring FLIM. There are time domain measurements that use a femtosecond pulse laser and a gated camera to determine lifetimes by their exponential decay using fitting methods. And as you've all heard, frequency domain uses a modulated excitation source and determines lifetimes directly by Phasor analysis. The key is that these are measuring the same events, they're measuring the same underlying physics, they only differ in the way you analyze that data. So we use frequency domain FLIM, and I know you've heard a lot about this. We use a modulated light source to excite our sample. And then we measure the emission characteristics. So we know precisely the waveform going in, and we can measure precisely the waveform coming out. And again, as you all know, because of the lifetime of the excited state, there's going to be a slight phase delay in that emission waveform. And there's also going to be demodulation of that emission waveform. And we can measure these both precisely and obtain a direct measurement of the lifetime. When I think of phasor plots, I know Enrico thinks of these naturally. For me, it's a struggle, but I think of these as a simple geometric representation of the frequency characteristics of that emission waveform. And so what I want to show you here is the way I think about it is every point on this emission waveform can be plotted on this two-dimensional graph as a vector rotating around an origin in a complex plane. So we're simply mapping that waveform onto this two-dimensional plot. And so you've seen these already. These are the phasor analysis. Every pixel in my image was going to undergo a Fourier transform into the S and G components. Fluorophores with a single component lifetime are described by the modulation of the cosine of the phase and have a lifetime distribution that will always fall in this universal semicircle. The other key point I want to mention, this is actually a purified fluorescent protein on a cover glass. And this is a 256 by 256 image. There are 65,000 pixels in this image. The lifetime characteristics of every single pixel in this image is represented on this phasor plot. This happens to be purified turquoise 2 that has a lifetime of 4.5 or around 4.5 nanoseconds and a single component lifetime. This happens to be purified cerulean. Cerulean actually has 2-component lifetime, and you'll see immediately that the lifetime distribution falls inside the semicircle. And this tells you right away that this is a multi-component decay. And the distribution of all 65,000 pixels falls inside the semicircle with an average lifetime of around 3 nanoseconds. So the beauty of the phasor plot is you instantly know what you're looking at. You instantly know when it falls inside the semicircle that it's multi-component. And of course, FRET will always be multi-component. So you expect your FRET distribution to fall inside the phasor. And so the other key point, I know Enrico made this again, is that phasors require no fitting. They're a direct measurement of the excited state events, and they are a direct measurement of things like FRET. So to show you how this works, we adopted an approach that Steve Vogel's group at NIH have used extensively, and that is to generate a series of FRET standards. So our preferred donors are in the turquoise family. We like using Venus as our acceptor. And so what we simply did is we took turquoise and Venus and we connected them through progressively longer linkers. And we again took Steve's example and we made a version of Venus that doesn't fluoresce, it doesn't absorb, it doesn't emit. What it is it's a mutation of that chromophore at that tyrosine position. And this mutant, as I mentioned, doesn't absorb and doesn't emit. So it still forms, it still has structure just like this, but it cannot fret. So that's our donor alone control. It's a very important control for these types of studies. So turquoise 5 amino acid amber is our donor alone. Turquoise 5 amino acid Venus is going to be our highest FRET, our maximum FRET standard. And then by inserting progressively longer linkers, we have decreasing FRET efficiency. And these are very useful for us to calibrate and measure how good our system is working. And so now this is a living cell that's expressing turquoise 5 amino acid amber. And remember, this is our donor alone control. It falls directly on the semicircle, telling us it's a single component decay. It has a lifetime of 3.8 nanoseconds, which is the correct lifetime for turquoise. This is turquoise 46 amino acid Venus, a much longer linker. You can see that the distribution is shifted to the right. You can see it's also moving inside the semicircle, indicating that there's more than one component. This has a lifetime of around 3 nanoseconds, which gives us a FRET efficiency around 24% for this particular standard. And then this is turquoise 5 amino acid, our highest FRET efficiency standard. This has a FRET efficiency down around 2.2 nanoseconds, giving us a FRET efficiency of 43%. So these standards are very, very useful if you're interested in doing these types of studies to make sure your system is optimized for these measurements. And so, as I mentioned, we're interested in looking at protein interactions in the living cell nucleus. And so we are interested in a transcription factor called the cat enhancer binding protein 1 alpha. And this is an unusual transcription factor. It was cloned in 1989 by Steve McKnight's group and was recognized that it binds to heterochromatin, which is an unusual behavior for a transcription factor that's important for turning on genes during differentiation. So what you can see here is the nucleus of a cell, and this is a mouse cell, and mouse cells package their heterochromatin in these discrete islands, and this is centromeric heterochromatin. You can see EBPa alpha preferentially localizes there. And if we do away with its transactivation domain and just keep the DNA binding domain, it goes exclusively to these regions of heterochromatin. So it actually is a very useful probe if you're interested in looking at FRET inside the nucleus, because it localizes to these regions of heterochromatin. It's an obligate dimer when it's bound to DNA. It allows us to detect interactions of these transcription factors when bound to DNA inside the living cell nucleus. So we're using FRET-FLIM initially to identify the dimerization of CEBP alpha. So this is an experiment now where we have CEBP alpha, just actually the BZip domain of it, tagged with turquoise. This is our donor alone. This is our donor channel and a FRET efficiency map. And what you can see is that the lifetime is around 3.8 nanoseconds. So it's unquenched donor bound to DNA. If we then co-express it with a yellow fluorescent protein, Venus, tagged to the BZip protein as well as turquoise, we now have the acceptor channel, the donor channel, and the lifetime map. And you can see the distribution of lifetimes has shifted to the right, moved inside the semicircle, indicating FRET, showing us that these proteins are indeed dimerized. What you'll also notice is we really only detect proteins dimerized in heterochromatin. The protein outside those regions does not FRET. But of course, we can get more information from this. We can actually use each region of the heterochromatin within a single cell nucleus as a region of interest, and we can make measurements in those regions of heterochromatin. But there's a big caveat here. We're independently expressing our donor and acceptor. And so every transfected cell is going to have a different acceptor to donor ratio, and that's going to influence the FRET efficiency. So measurements of donor lifetime to measure, if we're going to use donor lifetime to measure FRET efficiency, we have to account for the donor acceptor ratio. So if we co-express these proteins in a 1-to-1 ratio, what we expect is 1 out of 4 would be donor-donor and would not FRET. 2 out of 4 would be donor acceptor pairs and would FRET. And of course, 1 out of 4 would be acceptor pairs and they would not FRET either. So only these pairs are going to FRET. So we have to know what that donor acceptor ratio is in order to measure FRET between these proteins. And so we use FLIM to measure FRET efficiency. We measure it in multiple ROIs for each cell, so multiple regions of the nucleus for each cell, and we measure many cells. And what we do is we determine the acceptor to donor ratio in each cell. So each point on this graph is an individual cell. We measure multiple ROIs for each individual cell. So the spread here indicates the spread in FRET efficiency for the cell and the spread in the acceptor to donor ratio for the cell. And then we simply plot FRET efficiency as a function of the acceptor to donor ratio for each cell. And what we have is cells that have a lot of donor, going to have low FRET efficiency. Cells that have a high acceptor, that donor population is going to be saturated. And so this is going to be the maximum FRET efficiency for that particular pair, in this case, the BZip domains of CEBP alpha. And so we're able to, by plotting this, to determine what the maximum FRET efficiency is for a particular pair of fluorescent proteins. So this is the dimerization of the BZip, the DNA binding domains of CEBP alpha. So of course, we're interested in asking who CEBP alpha is interacting with in these regions of heterochromatin. And so as I mentioned, CEBP alpha, this transcription factor important for development, localizes to heterochromatin, which is an unusual behavior for a transcription factor. These regions are usually associated with gene silencing. CEBP alpha is important in directing programs of cell differentiation, so it regulates genes involved in energy metabolism and other genes involved in differentiation, but it likes to bind to methylated DNA and heterochromatin. And so we were wondering, why does it bind to methylated DNA, and who is it talking to when it's bound there? And so there were a number of studies that indicated heterochromatin protein 1 alpha, HP1 alpha, which is a protein involved in forming heterochromatin, also was capable of interacting with specific transcription factors. And so we decided to use FRET-FLIM to determine whether CEBP alpha was interacting with HP1 alpha in these regions of heterochromatin. And so again, we use this FRET-FLIM approach. We have a Venus-tagged HP1 alpha, the full-length HP1 alpha. We have Cerulean3, which is a variant of turquoise, tagged to CEBP alpha. And this is an example of one of these cells. This is a lifetime map. And we use this FRET-FLIM approach. We measured many cells. We measured many ROIs in each cell. And then we plotted the FRET efficiency determined by lifetime as a function of the acceptor to donor ratio. And as you can see, we have a FRET between CEBP alpha and HP1 alpha that saturates at a FRET efficiency around 15%. So this is telling us that indeed we are able to measure the interaction between these two proteins using this FRET-FLIM approach. But the beauty of this approach then is that we can begin to change these proteins and ask what's necessary for these interactions. So for example, this was done with the full-length CEBP alpha. Remember just the DNA binding domains necessary to get it into heterochromatin. And so we could ask, does HP1 alpha interact with just the BZip domain of CEBP alpha? So this is cerulean tagged HP1 alpha. And this is in the presence of a red fluorescent tagged BZip. And you can see they co-localize in heterochromatin. And then when we did the FRET-FLIM experiment, this is the result we got. Again, a maximum FRET efficiency of around 15%, indicating that just the BZip domain was required for the interaction with HP1 alpha. So that tells us that getting CEBP alpha into heterochromatin was sufficient to interact with HP1 alpha. Next, we wanted to know what's required within HP1 alpha for this interaction. And there's this mutation that was identified in the chromodomain of HP1 alpha. That's when you mutate this position, what happens is HP1 alpha no longer localizes to heterochromatin. It's what is required for its binding to heterochromatin. And so we made that mutation. And indeed, what you can see is that this cyan labeled HP1, mutant HP1 alpha does not localize to heterochromatin. However, if we co-expressed it with the BZip domain of CEBP alpha, they now co-localized in heterochromatin. So this tells us that the BZip domain is able to recruit HP1 alpha to the heterochromatin. But remember, this does not bind by itself to heterochromatin. And then we did the FRET experiment and asked, does the mutant HP1 alpha interact with the BZip domain of CEBP alpha? And we find that they really interact very poorly. Again, suggesting that by disrupting the chromatin binding of HP1 alpha, we've dramatically impacted its ability to interact, in this case with the BZip domain, suggesting that both of these proteins need to be interacting with heterochromatin in order for them to interact properly and function normally. And so we determined that they only can interact if both combined efficiently to heterochromatin. So that's the end of my experimental result. I just wanted to comment a bit about what we think is going on here. So HP1 alpha serves to translate histone methylation into DNA methylation by recruiting the DNA methyltransferases. And so there's an increasing evidence that HP1 alpha has a lot of functions in maintaining genome integrity, in regulating gene transcription, and regulating DNA replication and repair. So it's a very important protein. Our studies indicate that this transcription factor important for differentiation directly interacts with HP1 alpha when it's bound to heterochromatin. And remember, CEBP alpha likes to interact with methylated DNA. And so what we think might be going on is that when CEBP alpha wants to turn on genes required for differentiation, it simply comes in and interacts with HP1 alpha, binds to the methylated promoters it wants to turn on, and disengages HP1 alpha from this complex, allowing chromatin remodeling proteins to come in and begin to remodel that chromatin and turn on these genes required for differentiation. So it functions to activate and turn on these genes in heterochromatin. And I'm going to stop there, and I'm very happy to answer any questions you might have.